welcome to everybody this is the third lecture in our series on light, and we're delighted to have today Mohab Ibrahim from Arizona to speak to us, and after that our own Alistair Nunn. So without further ado, what I'm going to do is I'm going to ask Betony, who has been with us since 2021 in communications. It's a postdoctoral, has a postdoctoral role in Francisco's lab in Stellenbosch, and Betony is going to make the introductions to Mohab. Betony.
Thank you so much, Geoffrey. I'm very pleased to introduce this third session of what is proving to be a really fascinating series. Today's speakers are, as Geoffrey said, Professor Mohab Ibrahim and Professor Alistair Nunn. I'm very excited about today's subject matter, which is the therapeutic use of green light. And the color green has become almost synonymous with living things, which is ironic because it is the light that's actually predominantly reflected in pivotal biological functions such as photosynthesis, which uses mainly blue and red light. So what is the physics of green light? Very briefly, as I'm sure you will hear much more from our speakers, green light falls in the middle of the visible spectrum of electromagnetic radiation, approximately 490 to 590 nanometers, depending on the source. And it has higher energy than red light, but it is not ionizing like UV light. And the therapeutic history of green light is difficult to trace because while it has been recognized since antiquity that nature has therapeutic benefits, this was more often than not the exposure to sunlight rather than exposure to nature's greenery. But more recently, there's been a focus on effects of Shinrin-yoku or forest bathing, which emerged as a subject of interest as early as the 1980s. There have also been some studies about the use of green light to mitigate migraines, and as we will hear from Mohab, fibromyalgia. And what is very interesting to me is that it seems that the focal point of green light seems to be the treatment of pain, whether this is physical or emotional pain, such as mood disorders and depression. And I'm very interested to hear about the possible mechanisms by which green light might have this effect. So without further ado, it is my pleasure to introduce the speakers. The first speaker, Professor Mohab Ibrahim, attained his master's and doctorate in pharmacology and toxicology from the University of Arizona, focusing on neuropathic pain. His MD from the University of Arizona College of Medicine, an internship in the Department of Surgery at the University of Arizona. It is residency in the Department of Anesthesiology, Perioperative and Pain Medicine at Brigham and Women's Hospital, and a fellowship in chronic pain management at Massachusetts General Hospital's Anesthesiology Department. He is a tenured professor of anesthesiology, pharmacology, and neurosurgery at the University of Arizona, and the director of the Chronic Pain Clinic, the Chronic Pain Fellowship at Banner Medical Center at the University of Arizona, and medical director of the Center for Comprehensive Pain and Addiction. He is also a physician with the Southern Arizona Veterans Affairs Healthcare System. His work and research focuses on managing chronic pain for patients and training medical students, residents, and fellows. He maintains an active research laboratory exploring gaps in our understanding of chronic pain with a focus on photobiomodulation. And his research interests include the biology of pain and neurodegenerative diseases and discovering new methods to control them with an aim to translate animal research into clinical treatments. Our second speaker for the day is Professor Alistair Nunn. He is Director of Science for the Guy Foundation. He is a PhD biochemist who began his postdoctoral scientific career studying how cells undergo programmed cell death at Hammersmith Hospital in London. He continued his research in Edinburgh before moving into industry and worked in clinical safety and medical communications while continuing his academic relationships. He set up his own company in 2005, Broadman Science, so he could act as an independent scientific advisor. From 2010 to 2015, he was supported by GW Pharmaceuticals in academic positions at Imperial College and the University of Reading. And he is visiting professor at the Guy Foundation Quantum Biology and Bioenergetics Laboratory at the University of Westminster. Alistair's interests, he is primarily a theoretician specializing in mitochondrial function and the aging process and what actually defines health. His research interests include the origins of life and evolution from the quantum and thermodynamic perspective, and he is author on more than 20 scientific papers and has written chapters in 2 books. And with that, it's my pleasure to hand over to Mohab.
Thank you very much, Bethany. Allow me a moment to share my screen. First, I would really like to extend my deepest gratitude to this extraordinary opportunity and distinguished privilege to address such a esteemed assembly. And I got a sense of this earlier today. And this is a subject, though seemingly novel, actually holds profound intrigue. The exploration of how diverse colors may alter biological functions through exposure through the visual system. I'm also sincerely honored to express my heartfelt thanks to the entire Guy Foundation, with special appreciation to Nina for her support and guidance through this process. It's with great pleasure that I now share with you some of our pioneering work on green light therapy. Specifically, I will be shedding some light on pain and the bright future of green light therapy. As a disclosure, the green light therapy is actually patented by the University of Arizona, but the University of Arizona created a mitigation plan to address any conflict of interest, which includes independent monitors to review the data and results and reports on a semi-annual basis to the ethical committee at the University of Arizona. So what I would like to share with you today are mainly 2 things: appreciating the biological effect of green light therapy on pain modulation, and shedding some light on the biochemical and cellular effects of visual exposure to green light therapy. So, as I was preparing to address the subject of pain, please let me first try to comprehend its essence. Pain represents a distinctive and universal human experiences. It's capable of manifesting as both a beneficial signal and a profound challenge, an enigma really worth of our exploration. So what is pain? It's defined as an unpleasant sensory or an emotional experience associated with or resembling that associated with actual or potential tissue damage. And that's the official definition of pain. This experience is deeply personal, shaped by the individual interpretation and the culture in which the person grew in. And the effective management of pain necessitates addressing both the physiological underpinnings and the emotional dimensions of it. So, pain is really a vast, multifaceted topic. However, for the present, permit me to illuminate some of its far reaching impacts. It's really hard to quantify the emotional impact of pain and the suffering that people and their family experience. But if we want to put this in a numerical value, maybe it's prudent to see how much it costs the economy. So, at least in the US, on annual basis, chronic pain, which is only a subsegment of pain, costs over $600 billion annually. To put that in perspective, pain costs the economy more than heart disease, cancer, and diabetes, or at least more than 2 of these leading conditions together. Again, that does not address the emotional or the suffering component, but at least it puts things in perspective. And since pain is universal, humans and other species have been trying to manage it since the dawn of history. Of course, management was as wild and imaginative as the human mind can be. For example, this is a vintage picture that I actually found online in which I'm going to do my best to describe it. Someone is having, I think this is headache, and the treatment was he inserts his head in some kind of metal vessel that I really hope was well constructed, while a very kind looking gentleman with a sledgehammer is tapping the metal vessel. And the idea is to generate some kind of vibration. It was referred to as vibratory therapy. And there are 2 young ladies in the background. They seem like to be saying a prayer or something. Well, we came a long way since then. And just with other things, we start very slow and then we advanced. For example, this is Dr. Ralph. This was how anesthesia was administered in the past. thankfully, things have changed a little bit, and now anesthesiology is administered in a different way. Same thing with the pain medicine. Right now, we have plenty of tools to manage pain, whether they are pharmacological or interventional. And this is just a partial list to show some of the classes of medications we use and a really short list of the type of procedures we apply in the clinic, since we're no longer allowed to bring sledgehammers to pain clinics. Lawyers don't like that. So for that, we really need a different approach or a complementary approach, because these tools, despite how numerous they are, they don't always work for everyone. And some people, some patients, they prefer non-pharmacological or non-invasive methods, which really started to ignite our imagination and our thought process. What can we do to address this segment of patients? So generally speaking, to manage pain, we need to modulate the pain center in the brain or the nervous system. And in fact, that's what medications do. It's better life through chemistry. But what if we can modulate the pain center using external energy? So all we really need is to modulate the pain centers or to activate them or inactivate them. How we do that is just a matter of methodology. So, what if we can use an external source that's non-invasive to affect pain, modulate pain? So, being a physician scientist, one of the things we do is to find challenges in the pain clinic and then try to take it to the lab and come up with solutions. And pain was one of them. So, we need to find a way to access the brain through an external source. And one of the possible routes is the visual system, because we needed an entry point that must have direct or indirect neuronal connection with the brain. So the question is, are there neuronal connections, whether they are direct or indirect, from the eye or the retina to the pain modulating centers in the brain? And the answer is yes, through some of our work, but mostly from independent laboratories around the world. Uh, they mapped neuronal connections that starts from the retina and travel all the way to the pain modulating centers. And I'm not gonna really spend a lot of time delineating these nuclei in the brain. So at least the theoretical ground is there. That is, there is connection between the retina and the pain modulating centers in the brain, which helped us formulate our first hypothesis, which is activating the visual system using different wavelengths of light will eventually modulate the pain centers. So we set to go with our first preclinical experiment with rats. And what we did was a very simple experiment. We got some plexiglass cages, we acclimated rats in them, and we started exposing them to different wavelengths of light, which corresponds to different colors. And we wanted to measure the amount of stimulus it take them to say, or not to say, but to infer that they are uncomfortable. And what we did was to place a source of heat under their paw, and a normal rat, after 20 seconds of that heat, will just move away. And once they move away, the heat immediately terminates. So the rats are free moving. If anything bothers them, they will just walk away. And we tested this with several types of colors, from white as the control, to purple, blue, green, yellow, orange, red, and infrared. And what we noticed with blue and green lights, there was an increase in the threshold, which means it took more stimulus or more time for the rats to sense discomfort and move away. Interestingly, with red, we saw the opposite. It lowered their threshold. So rather than moving away at 20 seconds, now they're moved at maybe 15 or 12 seconds versus green light and blue, which increased from 20 seconds to actually almost the cutoff line, which is around 32 or 35 seconds, because we did not want to induce tissue damage. So we now know it, and we repeated this experiment several times. And the question was, now we know it works in rodents, will that work in humans? So, because if it's a non-invasive, low-energy approach, it was not hard to get permission from the University of Arizona to conduct our first clinical trials on two cohorts. One was fibromyalgia, and the other was migraines. And I chose these two cohorts because these are some of the most challenging conditions that we face in the pain clinic, mainly because we don't really fully understand their pathophysiology. We don't know why some people develop fibromyalgia. We don't know why some people develop migraines. So, because we don't know the pathophysiology, all we can do is manage the symptoms, or at least try to. So, with these clinical trials for fibromyalgia, what we noticed is that white light, which we used as a control, really had no effect on their pain score, which we measured from 0 to 10. So the average group, their pain was around 8 to 9. When exposed to white light, there was no change. However, when they were exposed to green light, their pain dropped almost by 50%. The intensity of their pain dropped by 50%. And we saw something similar in the migraine group, which is the panel on the lower part of the slide. Again, white light had minimal effect. It was statistically significant, but clinically irrelevant decrease in the pain intensity. However, with the green light, we saw a significant reduction in the intensity of the migraine. And I should mention that the patients we recruited were the patients that failed the conventional therapies that we have. And in addition to pain, we also wanted to measure the functional capacity, the quality of life, because I can tell anyone just sit on a couch in a dark room all day long and your pain will be better, but that's not a way to live life. So we wanted to also see their quality of sleep, which was improved with green light, and also their ability to exercise, their ability to work. All those improved. The frequency of the pain episodes and flare-ups, those decreased significantly with green light. One of the things which I never understood is the all admitted to having more desire to do house chores, which if personally, if I was sick, I would really milk it and try to avoid house chores at all costs. But I think they were very excited to be able to perform the regular things that they missed doing. So we knew that at least for green light, we were able to control chronic pain. But can we also address other types of pain, specifically post-surgical pain, which is acute in nature? So we went back to the lab and we used a surgical pain model, which is making an incision in the hind paw of a rat. And that typically cause both mechanical and thermal sensitivity for about 48 hours or 48 hours to maybe 7 days. And what we noticed is that pre-exposing rats to green light decreased the mechanical sensitivity for acute pain in both males and females. However, for the thermal sensitivity, only males experienced better results. Female rats had no effect. So that was a little bit intriguing that there is a sex difference in the response. So we wanted to look a little bit into the mechanism. So we collected segments of the spinal cords and we started looking at the endogenous opioids, and there are three: the beta endorphins, the proenkephalins, and the dynorphins. And these are the natural peptides or molecules produced by mammals to control pain, and they have other functions. And what we noticed is in both males and female rats, there was an increase in the beta endorphin and the proenkephalin. However, the amount of increase was greater in males than that of females. We also wanted to look at the receptor because, after all, I am a pharmacologist. Because in order for a molecule to work, it typically needs to bind to a receptor to produce some kind of intracellular changes. So we looked at the opioid receptors in the body. There are three, possibly four, that still being characterized, but for sure we know there are the mu opioids, the delta, and the kappa. And we looked at the structure or the expression of all these receptors. And what we noticed in males is that there was an overexpression of the mu opioid receptors when they were exposed to green light. Females, female rats, they did not have as much increase in the expression. We did not see any difference in the delta or the kappa between males and females. There was no increase. So what that told us is, for some reason, male rats increased both the endogenous opioids molecules or peptides and also the receptors that are needed to manifest the activity of these peptides. And maybe that's an explanation. It's not the explanation, but it can be an explanation of why male rats responded better than female rats. Interestingly enough, both migraine and fibromyalgia, women are more predisposed to developing these conditions, almost 9 to 1 compared to men. And in fact, the clinical trials we published, I think we had maybe a total of 3 or 4 men, and the rest of the cohort were women. So, but we also know, we recently published that nociceptors, or the nerve fibers that mediate a painful or noxious stimulus, differs from male and females. And we're still looking into why that is. And this work was done actually by Dr. Frank Poreka. He pioneered this work and he's looking more into it. So hopefully we'll have more on that part of the story. So we also wanted to see what happens to the cytokines when rats are exposed to green light. So we collected some of the cerebrospinal fluid, CSF, and we looked at 2 components, interleukins 10 and tumor necrosis factor alpha. IL-10 is anti-inflammatory. So the more of IL-10 we have, the less inflammation we get. And what we saw with green light exposure in rats is that the more we expose them, the more IL-10 they produced in the cerebrospinal fluid, suggesting that green light act as an anti-inflammatory. On the other hand, tumor necrosis factor alpha is associated with inflammation. And what we saw with green light is that it decreased the levels of TNF alpha in the cerebrospinal fluid. So green light acted in almost in 2 ways, increasing the anti-inflammatory component and decreasing the inflammatory component. And that's why, or maybe an explanation to why it was so potent. And there is a slide I did not show you in post-surgical pain in rats. We wanted to compare green light, the efficacy of green light, to NSAIDs like ibuprofen and opioids like morphine. And in fact, green light in the surgical model in rats was as potent as morphine. So we looked at the cerebrospinal fluid. Now we wanted to know, where is that change coming from? Is it the spinal cord? Is it the brainstem? So we collected segments of the spinal cord and the brainstem, and we wanted to see where we are seeing these changes. And for IL-10, actually for all of them, it was not in the spinal cord. It was in the brain, or specifically the brainstem. So with green light exposure, there was an increased expression of interleukin 10 or IL-10 in the medulla. There was decreased expression of tumor necrosis factor with green light exposure. And also we decided to look at interleukin 6, which is also inflammatory cytokines. We also saw a reduction in the interleukin 6. So again, green light relative to white light increased the anti-inflammatory cytokines and decreased the inflammatory cytokines. But then the question is, where are these cytokines coming from, especially in the central nervous system? There are the two main sources are microglia and astrocytes. So we decided to look at both the microglia and the astrocytes. And what I'm showing here is the data for the microglia. Now, generally speaking, the microglia exist in two states. One is activated, the other is inactivated. So when the microglia is activated, it indicates that there is a pathogen, there is a condition that requires the microglia to respond by an inflammatory reaction. So when the microglia is activated, the cell body increases in size and the processes or the extension, the arms, if you will, becomes shortened. However, when there is no condition to necessitate an inflammatory response, the microglia is what we call a scanning phase. So scanning, the processes are long because they are looking to see if anything is going wrong, but the cell body is small because there is no need to synthesize any of the material. So we wanted to look at the microglia with white light exposure, which is on the left side of the screen. And this is a video, I'll run it in just a second. And when the microglia is exposed to green light. So what we noticed is that with white light, the microglia had large cell body and shortened processes versus green light that had small cell body and longer processes, indicating that green light hindered the activations of the microglia. And that might be the reason why we're seeing less of the inflammatory substances being released. So we quantified the video that I just showed you. And what we saw is that green light significantly decreased the size of the cell bodies and increased the volume of the processes, turning the microglia into a scanning phase, where the control condition, which is white light, really did not alter the activated state of the microglia. So what that means is green light through visual exposure enabled us to modulate the neuroinflammation through modulating the microglia and the astrocytes. And we have similar data to astrocytes. We looked at it through immunohistochemistry, not 3D modulation, but the results were the same. So what we can conclude from this is that green light exposure resulted in the deactivation of the microglia or hindering the activation of the microglia, thus decreasing the neuroinflammatory environment in the central nervous system, resulting in changing the milieu of inflammation that's typically seen with either acute or chronic pain, such that it alter the balance towards less inflammatory environment. This work and some of our work was replicated all over the world. So that's always a pleasure to see that people are able to replicate the work. And actually, I should have updated the slide. There were recently published articles as well looking into the effect of green light. And what I talked about is green light through the visual system. That also does not take away from other wavelengths through the skin. So there are two routes of entry. One is through the visual system, which what I talked about, but also through the skin. And that's a completely different topic, but in general, red and infrared has better penetration through the skin that can also alter the inflammatory components, and can have similar responses in animals and humans. And one of the questions I was asked is, why not continue with blue light? Why did you focus on green light? Well, one of the things that chronic pain patients or patients in pain generally complain of is disruption of their sleep, because pain really can affect their sleep. And pain and sleep are intimately connected. One can exacerbate the other. And unfortunately, it becomes a vicious cycle where pain disrupts sleep and disrupted sleep aggravates pain further. Well, the problem with blue light is that it can affect the release of melatonin. It does something called phase shift. So melatonin start being released from the pineal gland later on. For example, if the natural circadian cycle for a person around 10 p.m., for example, that's when start to get sleepy, because that's when melatonin, their body was conditioned to start releasing. But if they are exposed to blue light, then rather than being released at 10 o'clock or 10 p.m., then it start being released at 11 or at midnight. And that's why some of the phones or some of the computer screens, they have a sleep mode. What the sleep mode does is basically take the blue spectrum out of the equation. That's why I stuck with, or one of the reasons why we decided to pursue green light um, rather than blue. That being said, blue does have its other biological function. In fact, one of the researchers and a colleague of mine at the University of Arizona, Dr. Scott Kilgore, he is researching blue light, and he found a lot of benefit. It's through visual exposure, a lot of benefit for PTSD. So pain management started with really interesting approach, but is it really that interesting? Because all they were doing at the time was applying external energy, trying to activate or modulate the pain centers. Maybe they did not at the time understand what they were doing. But this is typically, when I looked a little bit deeper into it, this was an expensive therapy. And you probably can tell by looking at the outfit the gentleman receiving the therapy is wearing. He's actually well dressed. So this is not a therapy that a lot of people can afford at the time. So no one is going to go through this therapy unless they actually noticed some benefit. So maybe we should go back or to the roots and really start looking and refining old methodologies that worked, but we did not know how it worked. So with that, I would like to acknowledge a lot of people that helped produce this wonderful work, and also the support from the University of Arizona, from the University of Arizona Comprehensive Pain and Addiction Center, from Oklahoma State University, the National Institute of Health, the Department of Defense, the Veteran Administration. All these entities actually supported and funded my research. And that's my wonderful lab. These are the people behind all this work. Some of them graduated. We got new members. So it's an ongoing project. We're very proud of it. And we are literally just scratching the surface.
Okay, well, thank you very much, Mohab. That was an amazing talk. I'm going to try and sort of do a bit of backfilling on the story of this on Greenlight, because when we were discussing setting up this lecture series, we were looking at different colours, and green came up, and I thought, well, what do we know about green? And having gone into the literature, it's turning out to be a really fascinating story. So I'm going to start by talking a little about the history, the galactic history of light and water, because water comes into this everywhere. I'll give a very brief history on photobiomodulation, why green, potential evolutionary tale, and some look at some of the mechanisms potentially underlying the green light therapy, and a few conclusions and some thoughts. Okay. Well, I'll start with this one. Probably most of you recognise this is of course the galactic centre, the Milky Way, and there seems to be the story of this may go back a lot further, and I think Bob will be familiar with this than we think. We are stardust, as everybody says, but are we also made of light and water, which I think is something which is perhaps more important than we realised. And certainly from the evidence, when I started looking into it, it seems that the story of water has been something which has been a bit hit and miss, but now with modern instruments, it seems that water is absolutely everywhere and may have even been around right from the start of the universe, and certainly seems to play a role, for instance, in the generation of how planets come to be in planet formation. And when we start to look around our own solar system, we're finding it absolutely everywhere, including on Bennu, who seems to contain brine with containing aromatic compounds. So this goes back a long way. And the other thing we also seem to find is that universe seems to be full of, or at least certainly when we're pointing out into other star system galaxies, there's an awful lot of chemistry going on out there too. And a lot of this is related to UV light, which seems to be driving the formation of these organic and what we say aromatic molecules. And so there's some very interesting stellar chemistry going on. And a lot of these seem to be potentially precursors to life. And I say aromatic because these, of course, are chromophoric molecules. Also, the geochemistry of the Earth seems to be essential for providing a stable thermodynamic condition to enable what some say is the how life started, which is the autocatalytic molecular self-organization systems involving the flow of electrons and hydrogen molecules, and to give more complex molecules. But of course, all of this has been only possible because of the quantum properties of water, which we talked about in one of our previous series. So it seems that actually life therefore appears to be a self-organizing dissipating system, which might be built on a chromophoric redox molecules. Potentially, these evolved in a geochemically active watery world where photons came from both geothermal sources and an external star. And these two sources are quite important. And here I've got obviously some kind of a stellar event, formation of planets, and a geothermal event. So there's a puzzle from this one. Did life require the deposition of aromatic compounds on Earth from extraplanetary sources, or were they all homegrown? We don't know. Puzzle 2. Which light source was most important? Internal, maybe from geochemistry and thermal vents, or external, or not at all? Certainly, just to remind everybody, water appears to be the most important metabolite in the body. It's involved in just about every single chemical reaction involving hydrolysis, etc. And it's very much, as this lovely quote from Indran said, water is the most perfect traveller because when it travels, it becomes the path itself. And when we look at, say, the citric acid cycle, the TCA cycle, water is involved in every step. So water is really important. And when we look at the water and then we look at some of these molecules which have evolved, and this is again just sort of jumping forward a bit, but many of the molecules in our body which are essential for electron transport, looking at NADH for instance, or FAD, FMN, and cytochrome C oxidase, including ubiquinone, are all chromophores, and they can absorb photons of light, and this alters their electronic state. Even DNA, and DNA is very sensitive to light. But we got into this, and then Geoffrey remember this, when we started to study, for instance, why is it that many natural products seem to work as medicines? And one of the paths we followed was, of course, we started to look at their structures, and many of them are aromatic, and they have phenolic components. Now, the literature will tell you that these compounds originally evolved when plants came onto the land to act as sunscreens, but we now know nearly all of these molecules are involved in stress resistance. And this seems that they have a pleiotropic effects. And a lot of this is simply dissipating energy. And one of the things they will have obviously is some very interesting quantum properties, and certainly related to pi electrons. One of the things that we've kind of suggested a while back, and I don't know how realist this is, but could these molecules themselves be important in, say, how we dissipate energy systems? And now we talk about autocatalytic networks, but we can also see that light can generate types of chemistry which can lead to self-organization. But certainly when we fast forward to modern eras, we know that life has been using these chromophoric molecules all the way through from the beginning. And back to the old chemicals, the plant compounds, we know that these are, of course, important in stress resistance. And this led us down a little bit of a path, and this was our best stab at explaining how natural products work. And this came from this idea that maybe they're important as not just cofactors, but prefactors that were around before life got going. But one of the interesting things about many of these molecules is that they are redox active. They also dissolve in membranes, and they're also affected by light. And of course, in biochemistry, one of the most fundamental homeostatic mechanisms in all life is the ability to maintain membrane fluidity. And of course, we're not quite sure when membranes evolve, but probably very early on. And if a membrane's too stiff, it doesn't work. If it's too fluid, it doesn't work. So biology is very good at keeping it in a very narrow Goldilocks zone. And it seems that a lot of these molecules can also dissolve in membranes and control this. And two of the things that control membrane fluidity is temperature and oxidation. So we've got this very interesting interaction between light, membrane fluidity, and certainly oxidation. And it seems that these molecules can affect all sorts of functions in a cell, ranging from mitochondrial function, ion channels, nuclear membranes, potentially, even potentially morphogenetic fields and bioelectricity. So there's some interaction going on here, which is very, very ancient. And one of the things that got us really interested in this, and this is where I was working with Stan, when we were trying to look at cannabidiol in live cells, because it fluoresces, and we were throwing it into some experiments and using a 2-photon, and we basically fried the cells. But one of the things that made us really realize when we started to study the absorption and the emission spectrum of the major molecules in life, they all overlap. Now, so you've got tryptophan that absorbs way down in the UV, then you've got NADH as you go up the wavelengths. But what's interesting is, of course, when they emit a photon at a longer wavelength, it often overlaps with the absorption of another. And the key point here is where we looked at this. If you look at, for instance, the excitation and emission of NADH, it absorbs around 350 nanometers and emits somewhere around 450, 475. But of course, it overlaps with FAD plus, which then emits at a longer wavelength. And of course, that wavelength is where porphyrins absorb. And what was also very clear that, of course, when we change the redox status or we shine light, their absorbance changes. There's a very close interaction between redox and light absorption. And is this surprising? Well, of course it isn't. Life evolved at a certain set of conditions. We've discussed this with Lynn actually about why life evolves and why it's difficult in space. But it's canalized to work in a certain Goldilocks zone. And so light probably has this role of being either too much or too little. And I've seen there's been some comments about biphasic responses on light, the dose effect that we're talking about. And there is a Goldilocks zone, so too much or too little is probably, we need to be just right. And of course, the other thing to throw in here before we get on to green light, of course, is biophotons. Now, this is not a new idea. People have been studying biophotons, and these are metabolically produced photons. Life is all about redox, and if redox goes slightly awry or changes, we get the production of reactive oxygen species, and these can generate light. So one of the theories behind biophotons, certainly, that metabolism is always producing low levels of photons. And certainly, if the cell is full of chromophores, this photon can then be absorbed by another molecule. So is this a form of photonic homeostasis? We don't know for certain yet, but it's something which would be really interesting if it's true. And if it's true, when did it arise? So moving on to photobiomodulation. Now here I've got a picture of the sun and two lizards, and it's well known that things like, for instance, cold-blooded reptiles, they have to absorb some sunlight to warm up. But of course, the theory that's also out there is maybe it's not just the heat, but it's also the absorption of light that could be doing something to their systems that enables them to function better. So how you dissociate the photon energy from thermal is obviously something we need to investigate, but this is not a new idea. And if we're going back and we're thinking about sunlight, of course, heliotherapy, which is sunlight therapy, is very, very ancient. There's evidence of it going on with ancient Hindus. Certainly in Greeks, they used to use light that they thought was associated with health and therapy, and this is why certainly some of the idea that the sun gods are here. But if we jump forward, to the time of Florence Nightingale, she realised that actually light could help, and certainly sunlight could help to accelerate healing of soldiers' wounds. Of course, it was Niels Finsen, as Geoffrey mentioned, it really took it to town and started to show that certainly you could use light to treat wounds and help healing. And in the 1940s, 50s and 60s, it was Britton Chance who really started to investigate the role of respiration and electron transport. And one of the things he discovered or is involved, and we came to really understand was a good way of looking at things, was that these chromophores inside, say, mitochondria could tell you what the electron transport chain was doing. And this sort of gave birth to the idea of photobiomodulation, but of course photoplethysmography, and the idea that you can study metabolism by using light. And of course, all of us, or most of us who are using Fitbits, Garmin's, etc., will look at the back of our watch and see it's producing green light. And of course, this is used to monitor your heart rate. But of course, other wavelengths are now being used, for instance, to measure your saturation of oxygen. So this is an old idea. It's coming together now, and it's some really interesting pieces of instrumentation. And we also know, of course, as mentioned, that light is obviously having healing properties. And it kind of leads us to this idea of quantum optical theranostics, as I call it, which is where you can detect but also treat with the same modality. Thinking about photobiomodulation and chromophores, these are the ones that are kind of accepted. Now, of course, photobiomodulation requires a chromophore. It's a bit like a drug requires a receptor. This is basic quantum mechanics 101. This is how quantum mechanics came into being. It's the interaction of light with matter. What's fairly well accepted that certainly in biology, cytochromes are one of the key receptors for light, and many of these are porphyrins based on both heme and things like chlorophyll. We also, and certainly in the series, we've been talking about cryptochromes, and these have cofactors like FAD, which, for instance, are important in circadian rhythms, but also potentially magnetic sensing. Then we've got the molecular chromophores, as I like to call them. Of course, these are individual molecules which also absorb light, and these are the aromatics I was talking about. And then, as Mohab mentioned, of course, there is opsins and there's trip channels. Now, I'll talk a bit more about the trip channels later. These could potentially be really interesting. We then got nanocluster water. Again, we've mentioned this briefly in some of our series about the idea of ordered water. What is the role of force fields in the water, electrostatic traction, etc. And then there are non-mitochondrial cytochromes, and there are things like cytochrome P450 and other enzymes which have moieties which can absorb light, although they're not really been investigated in great detail. Which brings us to, can we simplify some of this? We've been thinking about this for a long time. And of course, we're mitochondrial people. And certainly if we think of the membrane, it's got all sorts of proteins and things in it, some of which can absorb light, and many of them which absorb light can also alter the flow of calcium and other ions into the cell, and the trip channels in particular. But of course, also below the membrane, we have things like cytoskeleton. As Philip obviously talks about the role of that absorbing light and superradiance. But within the mitochondria, we of course got the cytochromes, and these I think are particularly important. But one of the things all of these interact with is, of course, redox and calcium signaling. And because mitochondria are central to calcium signaling, they have all sorts of roles. And this is a little diagram we produced a couple of years ago. Now, this used to say cannabidiol. This also now says light. So low stimulus, for instance, of either calcium into the cell or activation of the mitochondria, can cause shifts in calcium signaling involved in the mitochondria. And low doses, for instance, can cause a mild adaptation. You can cause, for instance, an induction, say, mitophagia and or mitochondrial turnover. Too much, of course, can introduce apoptosis. And this is your biphasic effect. And mitochondria are very good at buffering calcium and modulating redox. So you can see how this light can translate into a calcium or a signal within the cell. But of course, what we're really interested in here is green. So many of you who've seen Blackadder, of course, remember Purist Green. I have a history with this. This is a series on the BBC TV with Rowan Atkinson, and they had Lord Percy was a not very clever man, but he was trying to do some alchemy and generate gold, but he ended up producing Purist Green. And as luck would have it, in one of my experiments, at the same time this was on, I actually managed to produce Purist Green from mucus, actually, but that's another story. So what do we mean? I think, as Mohab said, green is this obviously this wavelength between 5 to 600 nanometers. But why is the world very green? Well, mainly because plants don't absorb green very well, and although it does play a big role in plant function. So they absorb, the green leaves absorb red light, blue light, and reflect quite a lot of green lights. That's why we see everything in green. But here, this is where the water comes into it. And this gets back to Bob Fosby's thinking, etc. Water has a very specific absorption and dissipation of water, of light. And we know that if you look at the ocean, infrared light gets mostly absorbed near the surface, red light goes a bit further, but green and blue light go a long way into the ocean, 30 or 40 meters, depending on the intensity. UV light also goes in, but of course, the shorter the wavelengths, the less it gets absorbed. Now, the interesting thing about this is, of course, that red light and infrared light seems to warm the water, but UV light, especially at high intensity, can cause photolysis and photolyase and produce reactive oxygen and ozone. So there's another piece of chemistry going on because it's ionizing at the other end of the spectrum. Is this important? Well, may well be, because one of the theories of the origins of life is that UV played a very big role. And very early Earth, it may be that there was so much UV hitting the surface of the Earth because there was very little blocking it and the sun was potentially producing more. Life could not have started near the surface, and this is a paper by Nossen suggesting that in present day time it's quite UV protected, but the early Earth probably wasn't. And this might have big implications to where life may have started. And this is just showing how far shortwave light goes into the sea. So it's 305 nanometers, not very far, but 380, so near UV does penetrate quite well into seawater. And this again is just a better description of water. And this is where it's so fascinating. And again, Bob's talked about this, but you look at UV, water absorbs it very well and ionizes it. Then there's this little transparent band where we have visual. But as we go into the red, of course, it gets much less. It gets absorbed and it doesn't penetrate terribly well, but it does seem to absorb, and then you can increase the temperature of water, as we said. The other thing that may be going on here, of course, is that it may be doing something with quantum resonance. We don't know. It's not very. We don't really understand this yet. But there's two distinct things. But the clear thing here is we've got this little visual window. Now, of course, we're thinking about green, and one of the things when we think about green, of course, is the evolution of chlorophyll. And what we know is that chlorophyll and bacterial chlorophyll, they don't really absorb very well in the green. That's why they look green. But interestingly, purple bacteria and haloarchaeal bacteria do absorb very well in the green. Have we ever thought why this is? Well, let's just start at some of the clues. Humans are very good at picking up green light. We have a very high quantum efficiency in our cones for picking up green light. That's why it appears so bright to us. Opsins are clearly clear in this. Now, the other things that absorb light very well, green light, of course, is bacteria, purple bacteria. So we've got a commonality with these two. And in fact, as I said, green light is reflected by plants, but actually quite a lot of it is actually absorbed. It's quite high energy. But it's more efficient at absorbing blue and red. And this is just a diagram showing the absorption of some of the light harvesting complexes in the bacteria. But okay, I'll just draw your attention to this diagram here. This is where the green light of rhodopsin comes in. And these guys can absorb red and near-infrared light and some blue light, but also very much this green light. There's a clue here. And why is this important? Well, this is a bit of speculation, but I think it may be telling us something. This is a paper that came out several years ago, and this is a theory by De Saar and Schreiterman that actually on the early Earth, most of the light you'd have seen would have been purple, not green. And this is because the purple bacteria were really thriving and absorbing green light. And this was before oxygen came along. But when oxygen started to come out and was produced, say, by early forms of photosynthesis, this led basically to a dip in green light. And so when evolution kicked in with the modern photosynthesis, things evolved either side to pick up light from either the red side or the blue side, but not so much the green side. And so this theory is that a green world evolves after oxygen appears, as anoxic photosynthesis heavily absorbs green light. So newer oxygen tolerant systems would have evolved using available light wavelengths. And this is now being taken up by the Astrobiology Brigade, and they're saying rather than looking for, as we point our telescopes out into space, looking for exoplanets and potential evidence of life, we shouldn't be looking at green planets, but we should be looking at purple ones. Now, I think that's quite an interesting thing. And actually, the biochemistry, there is some hints of biochemistry here, because the early forms of biochemistry could have produced retinol, which can absorb in this area, whereas more modern TC, well, I say more modern, it might have come across at the same time, we don't fully know, but could have led to chlorophyll, which has a more blue or a red absorption. But the key thing here, of course, this is all very much linked into electron transport. Now, the evolution of electron transport chain, and this evolved very early in life. We don't exactly know when, but this is how biology uses electrons to generate a proton gradient, or at least capture energy. Some of the earliest proteins that we can divulge from phylogeny studies and going back through genetics, which could be very old and predate what they call the last universal common ancestor of all life. One of them is ATP synthase, which I'm sure Wayne will be familiar with. But there are other things occurring at the same time about how to use some of this energy and adopt some of this energy, especially when they started to metabolize other electron donors. And these involve flavins. And this suggests that there was a very tight integration with these molecules which can aromatics, which can absorb light. So how far back and when that goes, we don't fully know, but it suggests something quite interesting. And the reason this gets really interesting, if we go back to our UV story, it suggests that very early on the very early Earth, UV may have been too intense and it just destroyed everything on the surface. But our old friend, the alkaline thermal vent, and these are the deep sea vents which come in these subjugation plates, and at the moment seem to be one of the strongest candidates for a life start. And one of the key things they do is glow. And this is where you've got hot upwelling water containing hydrogen meeting seawater. And certainly in the early days, this could develop autocatalytic networks, but they glow. So they're producing light. And there's this lovely theory by Martin, and he's one of these folk who are very much into the origins of life. He suggested that photosynthesis could only have started in thermal vents, not near the surface. So this tells us something very interesting about the type of molecules that may have been absorbing light and then start to utilize that light to generate proton gradients. So I think we have to think slightly differently. Now, I'm going to jump forward a few billion years here and come to the origins of the cytochromes. And there's a reason for doing this. In the 1880s, they were trying to, certain folk like my mum were trying to identify the respiratory pigment in muscle. They were trying to work out, how does muscle work? How does all of this work? But it wasn't until Keelan in the 1920s, he actually identified pigments involved in respiration and called them cytochromes. And he called them cytochromes because all cytochrome means is a pigment in the cell. And he identified 3 in particular, A, B, and C. And these absorb at 605, 565, and 550 nanometers. Of course, this is going into the green. And here is I took it from his original paper, and this is where the green is. So these molecules, which are inside, clearly are components of electron transport, so and therefore inside your mitochondria, are quite good at absorbing green light. And what we know is, of course, and going back to the word of Britain chance, we know these can allow us to understand and follow how the electron transport chain is functioning. And the key thing is that each of these complexes has a mix of these cytochromes, and of course plants are the same thing. And we know that light alters these, and there's just several papers showing that as you alter the redox status or light, you can change the way these things absorb light. Which leads us back to our lovely friend, the mitochondrion. Now, there are theories here, and I'm sure Douglas will probably have alternatives, but one of the theories that was knocking about a long time ago, and I first thought this was a great theory by Cavalier-Smith, that the original endosymbiotic event, when a bacterium got together with an archaean to form the modern eukaryote, was that the alpha protobacteria was actually a purple non-sulfur bacterium. I don't know how accurate that was, but has lost all its photosynthetic components. But when we look at mitochondria, their color, the muddy brown, apparently they are muddy brown, largely comes from its cytochromes. Now, complex one, the first one, doesn't contain the cytochromes, but it does contain FMA and iron sulfur clusters. And we know this absorbs very well in the blue. And this is something which has been quite well described, which affects its redox status. Complex 3, which is a really interesting one called cytochrome C reductase, contains cytochromes, and it contains B and C. And these can absorb all the way from 407 nanometers right up into 605 nanometers. And they've got a peak around the green absorbance. Complex 4, which is the one everybody's interested in terms of red and infrared light, contains the A cytochromes and absorbs again from 420, but all the way up into the red and infrared. So we've got this very interesting mix of cytochromes. When, for instance, you alter mitochondrial function, and this is key, say, for instance, if you take a mitochondria and you treat it with an uncoupling agent, and so short circuit the proton circuit effectively and it oxidizes, you get a very big change in the absorbance, suggesting that there's a very close relationship between light and redox. So let's move on to the therapeutic. I think we're all fascinated by what's going on here. TRPV1. Now, Geoffrey and I are very familiar with this. TRP channels are fascinating for many things. They're quite old channels. They're ion channels. They mostly control calcium intake in the cell, but they're very important in sensing the cell's environment. And they cause an influx of calcium. And because they cause an influx of calcium, they can modulate mitochondrial function because mitochondria are one of the central controllers of calcium signaling. And they can hoover them up, they can hoover the calcium up. And as been suggested, we know that these channels seem to be coupled to opsins. And this has been shown by many people, suggesting that actually green light can affect through an opsin TRP channels. And there's quite a lot of evidence out there, many papers showing this. And I came across this paper by Wang, which showed quite an interesting thing, is that if you apparently stimulate these things with green light, you get a decrease in ATP, an increase in ROS, and a lowered mitochondrial membrane potential. This spoke to me, are we talking about mitochondrial uptake? Because we know that if you control mitochondrial function using components, you can control respiration quite tightly. And certainly many people looked at this, and they've looked at the trypsin opsins and suggesting, of course, this could be very important in bone repair and other healing properties. But of course, this does raise a very interesting point, and this is, I suspect, why this field is somewhat sort of mixed up. Of course, this effect will be modulated by what these channels may be doing in terms of, say, detecting things like capsaicin, chili pepper, other components of other phytochemicals and other ligands in the cell. But of course, temperature will have a big effect, and way opsins work is you get a cis-trans change when they absorb a photon. Now, I've looked very hard into the literature to try and find a bit more on this light thing, and I've only come across really one paper that seems to have gone in, and maybe Doug has got a few more on these, but there's only one paper that really gone to town and looking at the effects of light on different complexes in the electron transport chain. And they did this by looking at the light wavelengths from 379 nanometers all the way up to 1064. And they did this by using selective inhibitors of complexes in mitochondria. And what they showed was a very clear dose effect, which was related to the wavelength and thus its energy. And they certainly saw what we would call hormetic effects, which is a biphasic response, especially at shorter wavelengths. And this is just a diagram from their paper showing that the complex one is affected by near UV blue light and NIR, interestingly enough, but complex 3 is very much affected by green light. And red light obviously affects complex 4, which is the one everybody's interested in. So there's some very interesting dose responses going on here in terms of intensity. And as you would expect, short wavelength light has much more energy and potentially more damaging. Long wavelength has less energy. Looking at other papers, one of the problems, I think, again, is trying to find a common flux rate between these different papers and what's the intensity and dose. But certainly there's plenty out, and this is just a selection of the ones I found. Everybody seems to be focused on human adipocyte-derived mesenchymal stem cells, but this paper by Tomini suggests it was anti-inflammatory, which fits with what Mohab was saying. This other paper, giving a slightly different definition of the influx, showed that, interestingly enough, green laser light could inhibit cell proliferation and osseous differentiation, but it stimulated cartilaginous differentiation, i.e. it had different effects in different cells. Same thing again with another paper by De Silva, but they also showed this effect on osteoporosis. And interestingly enough, this one paper showed that they wondering whether green light could offset some of the signs of photoaging of UV. Again, showing some kind of interactive effect. And I know one of the questions was, say, about white light. Well, here we have an interesting thing. Light at different wavelengths is definitely doing something. Now, this is the paper I mentioned about complex 3. Now, this has come out quite recently. There's been a real resurgence of interest in the relationship between the various complexes in mitochondria and control of immunity. And they've seemed to have shown that LPS lipopolysaccharide can stimulate the production of ROS from complex 3. And this seems to, through various pathways, activate the production of IL-10. The suggestion here is, of course, that it now appears that complex 3 in particular is what they would call a metabolic checkpoint, that it controls immunity. Now, if this is going on with LPS inside macrophages, for instance, or any other cell, clearly green light, if it's going to modulate this thing, could do the same thing. Now, I wonder whether this is potentially something begin to explain some of its anti-inflammatory effects. That these people didn't look at green light. I'm just making a connection here between what happens, but this is a diagram suggesting this. This is quite a good paper. So if you want to see that, I can send you the details of these papers. So basically, green light controlling inflammation to quite a specific effect in complex 3 in the mitochondria. The other thing, dose is everything here, and we're very much we love the concept of hormesis, which describes, as Geoffrey said, Paracelsus' observations. Dose makes the poison. But actually, when you think about it from how life may have started, it's perfect sense. Stress, life would never evolve without some stress, and it would be biphasic. Small amounts of stress induce adaptation. Too much causes things to fall to pieces. And I've taken this diagram from a paper from Michael Hamblin's group. And this is they made this a long time ago and said, well, actually, if you look at light therapy, you've got the power density versus the time, and you can get stimulation and inhibition. So if you get these things changing, you're going to get different effects. And this explains, I think, why in the literature there is so much variability in the data, that we just haven't got this right yet. Right, sorry about the complicated diagram. So trying to summarize this, the green mechanism, one of the things we can start up in the corner here, if we got green light, say, affecting opsins and, say, trip channels, you get a flow of calcium into the cell. Now, this can go in a number of different directions. It can go into the endoplasmic reticulum. You can also get activation, what they call stop-operated calcium entry, but it also is taken up by the mitochondria. The mitochondria acts like a buffer and then sends out signals. So that can either induce adaptation through retrograde signaling, and potentially affect the unfolding response or the integrated stress response. There are numerous things that could be doing this. Obviously, the calcium ROS itself, but also metabolites, fumarate, succinate, etc. Epigenetics could be changing, and even metabolic photons. And critically, of course, and this is something which not everybody appreciates, is, of course, it also may depend on what state the cell is and where your mitochondria is. If it's near a nucleus, and we know they do this, it can alter nuclear function. But if it's near a surface of a cell, and maybe right next to the, say, the plasma membrane and the trip channel, you have a different effect. Or indeed, if they've undergone fission, or they're actually fused. All of these things, and we know that the state of the mitochondria and its fusion fission status has a big impact on calcium signaling. So that you can see how these things will begin to interact with each other. Now, then throw in an effect on, say, production of ROS from complex 3 or other complexes, you can start to see how all of this starts to integrate into a signaling adaptive system in the cell. And it's just to show this here. But interestingly, of course, you can have local effects. Now, we know that if you change all of these things, you get effects on glutathione, you get effects on NRF2, all these transcription factors in PGC1 alpha, for instance, involved in mitochondrial function. But certainly you can bring up FOXO1 and sirtuins, or FOXO3, and how these adapt to stress, but also the production of mitochondrial derived vesicles. And these are something which are coming out to be really important. The mitochondria can adapt slightly by reducing these vesicles. But of course, you might even have a systemic effect here. And we were talking earlier about the effect on the eye, but of course, if it was going into the skin, you might produce cytokines and extracellular vesicles, metabolites, and even down to the effect of nerve control. What happens if you shine a green light on your vagus nerve or something? These kind of things all start to come in. So we can start to see how there's multiple mechanisms, and up to and including other chromophores, which we haven't really looked at. I'll put NO here and IL-10 here as well. Clearly, one of the things that's going to be important here is the absorption of light, how far it goes. My suspicion is that complex life has evolved to block out the more higher intensity wavelengths, but be more more absorbent of the longer wavelengths. And certainly this could have a big effect on what we see. And what's interesting is there's evidence coming out in literature now that longer wavelengths, like infrared light, can protect against UV light and blue light. Now, whether this is a direct effect, for instance, on something to do with electron transport feeding back through this system, or on other things like the trip channels, or as suggested by Ludovica, you get this, and this is pure photophysics, and I don't pretend to understand it. Stan can probably explain it, but you get this thing called reverse intersystem crossing, where it affects the kind of actual absorption excitation status of a chromophore. But again, this is going to be something which is going to be where dosage is really important. But what I have to emphasize, if you look into the people talk about these things not being potentially damaging, high levels of NIR are damaging, as is green light. If you have the intensity too high, anybody that's looked down the end of a green laser will tell you all about it. You can have too much of a good thing. So you've got to have the right dosing. And this will also be affected, I suspect, by the cell state, its redox state, its metabolic reserve, how healthy is its mitochondria. This will have an effect on it, and potentially other signaling pathways. So I'm coming to the end here. It's work in progress. You can make a supposition that maybe because electron transport systems in life are predominantly based on chromophores, which is unlikely to be a coincidence due to quantum requirements, light will affect biological function. Spectrum of light that life will evolve under will have heavily influenced how it works, which in turn depend on the absorbance spectrum of water and circadian rhythm. So this goes back to water again. I've mentioned if life is based on fractal dissipation, but this is the idea that self-organization started very small and got bigger, so it's almost scale invariant. But where it induces the adaptation, where a local level, simply a molecular or a whole level of a species exposed to UV light, is something which we could look at. What we see and what we get depends on dose and scale. If the early Earth theory, purple Earth, is correct, then it might explain why plants are green and potentially why mitochondria are brown. early life would have been partly driven by evolution of the systems to dissipate the energy of the photons that could penetrate water. If the UV hypothesis is correct, it was going to evolve down here somewhere. If it did start in an alkaline thermal vent, it would suggest that their light spectrum would have had an influence the original autocatalytic chemistry and how it evolved and thus the electron transport chain. This still remains the big questions. Would aromatic chemicals that were produced by stellar chemistry be key in the origin of life, or were they all produced by the original geochemistry? So conclusions, I would say that from basic principles, green light is going to modulate metabolism. I think we've got plenty of evidence for that. It is likely that its effects are going to be modulated by other wavelengths. I think there's proof of this. What happens will depend on dose, the penetrance, density, location, morphology of mitochondria, where they are in the cell, type of organ and system, and even the organism overall homeostatic state, healthy or not healthy, robustness, for instance, metabolic reserve. And the underlying mechanism will depend on the chromophores interacting with both opsins and cytochromes probably certainly playing a big role, but there may be other things like modulating indirect modulation of mitochondrial function, verbal redox and signaling. And so really, I'd just like to finish on this slide. We've got two sources of light for our planet. We've obviously got the stellar version, but there's also thermal vents. Were these influencing the early forms of electron transport through photosynthesis or indeed other electron transport systems? If the purple Earth 3 is correct, this has some legs to it. Of course, this gave us the brown mitochondria and ultimately green Earth. And did light influence the evolution of electron transport chain, and was the depth of water dependent? There are some of these vents which are quite close to the surface. There's a field, and I'm not going to pronounce their name, somewhere near Iceland, which are within 30 or 40 meters of the sea surface. Was this important, rather than deep sea vents? But does this all hint that where life started? And I think this takes us right back to a very big moment. Anyway, thank you very much. This is the end of the talk. Maybe there's beginnings of much bigger discussion here. Thanks very much.